recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT, or TA, and GC, or CG base pairs is the same.